Okay, so it's my pleasure today to introduce Dr. William Valdar as our virtual speaker. So Dr. Valdar received a PhD in protein structure bioinformatics from University College in London, and also a master's in applied statistics from Oxford University. He completed postdoctoral research in bioinformatics and statistical genetics with Richard Mott and Jonathan Flint at the Wellcome Trust Center for Human Genetics, and then received a MRC Career Development Fellowship before transitioning to an adjunct assistant professor in the Department of Computer Science at UNC Chapel Hill. According to your CV, correct me when I make things, if I say something wrong.
So I moved to from the MRC Fellowship to become an assistant professor in the genetics department.
Genetics department. Okay.
And just as a courtesy, I became an adjunct in a couple of other departments as well.
Yeah, I saw that. So you then joined the genetics department as an associate professor in 2015, right?
I joined the genetics department as an assistant professor in 2009. I became an associate professor in 2015. Mm-hmm.
Okay. Yeah, and I guess you wear a lot of hats because you have a lot of appointments, including in the Department of Biostatistics, and as you mentioned earlier, Director of Graduate Studies for Bioinformatics and Computational Biology Curriculum.
Yes, I do a lot of things badly.
Dr. Valdar has also published 70 original research articles and is currently funded from diverse sources, including the National Institute of General Medical Sciences for development of statistical and computational methods to model multi-parent and genetic reference populations. So Dr. Valdar is an expert in statistical genetics and in the use of genetically diverse model organism populations. And his work has been critical for understanding trait genetic architecture and specifically modeling complex genetic effects like gene by environment, gene by treatment interactions. And I think his work has been really pivotal for the broader research community in terms of providing guidance and also tools to be able to leverage some of these genetic resources like the collaborative cross and the diversity out cross and heterogeneous stock mouse populations. So welcome, and thank you for taking time out of your busy schedule to talk to us today. And I just want to add really quickly, if you could just enter your questions in chat, and then we'll take questions at the end of Dr. Valdar's talk, and then transition to a Q&A period. So thank you and welcome.
Thank you very much, Megan, and thank you for inviting me. So I'm going to talk about modeling gene by treatment interactions in the collaborative cross and other genetically diverse replicable populations. And so first of all, I'm going to define what I mean by gene by treatment interactions, and I mean it very broadly. So by gene, I mean either an actual gene or a genomic region or a genome. And by treatment, I might mean a drug or some other therapeutic intervention, a toxin, an infection, or some other insult, maybe an environment or a diet. All of these things I'm just sort of broadly describing as a treatment. And so what I define G by T as is where the effect of the treatment varies with genetics. And the field of looking at this, it's really sort of multiple fields, but depending on what NIH institute you're writing your grant application to, you might call it pharmacogenomics, toxicogenomics, precision medicine, G by E, etc. But I'm just calling it G by T. And what I'm going to propose in this talk is a systematic model organism-based approach to G by T discovery that leverages genetic diversity, biological replicates, and powerful analytic strategies. And the implementation that I'm going to describe is with a collaborative cross, but it also generalizes to other replicable multi-parent populations and even to other genetically distinct replicable populations. So I'm going to start off by sort of introducing the collaborative cross and multi-parent populations. I'm not going to assume that many people in the audience know what those things are. And then I'm going to talk about some G by T concepts. And actually, I'm going to keep this talk, a lot of this talk will be at the conceptual level. Then I'm going to go through some G by T studies that that we've worked on in the collaborative cross and something called the diallel, and I'll end talking about power and reproducibility and how that fits into the kinds of studies and the populations that I'm going to talk about. So probably many people in the audience know about inbred mouse strains, but just for anybody who is a bit kind of hazy on it, so an inbred mouse strain, if you were to go outside and get a mouse from the forest and get another mouse, they would be genetically distinct from each other. They would be outbred, and then if you were to mate them with each other, then their offspring would be more similar to each other than the parents were to each other. So that, you know, you're more similar to your siblings than your parents are to each other, hopefully. And if you were to take those siblings and then mate them with each other, their offspring would be more similar to each other. If you were to keep on doing this, you would be sort of homogenizing the genome until, you know, after about 20 sib matings, the genomes of the animals would be inbred. And so all of the individuals at that point and their offspring will be genetically identical. And this has a number of experimental advantages that, you know, people in biomedical science who use model organisms take advantage of all the time. So you're essentially able to test multiple clones, and this means that you can phenotype more precisely. You can control for genetic background. You can replicate studies because you can literally redo the same experiment on, at least from a genetic standpoint, the same individuals. And that's a good way in which studies can kind of build on each other. Also, if you're doing multiple things on a single strain, and the rest of the community is also doing things on that strain, then there's the opportunity for the genome of that strain to be well characterized, and you can just continually take advantage of that. So if you're in my world of mouse genetics, and you often will represent an inbred strain by sort of two equally colored lines representing genetically identical chromatids of a chromosome. And so what I'm representing here is two inbred strains, inbred strain A and inbred strain B. So they're genetically different from each other, but they're individually inbred. And you might find that there are two strains that are different in an interesting way for a phenotype that you are studying. So, for example, maybe inbred strain B, those mice are extremely anxious, and inbred strain A, they're very self-assured. And then you might ask, well, these are genetically different, they're phenotypically different. How can I figure out where the, what it is about the genomes that's causing this difference? And the standard way to do that would be QTL mapping. And so what this will involve is crossing these two strains. And first of all, you'll create these F1 individuals who are sort of hybrids of both. And then you create an F2 population. And the F2 individuals are all distinct. You would also expect them then to be to have a range of anxiety, some of them being very nervous, and some of them being very self-assured, and a big kind of distribution of that. And you know that if you were looking at the variation among individuals at a location that was not involved in anxiety, then you wouldn't really see a pattern. If you were kind of to take the animals who are AA at one location and AB and the animals that are AB and BB, you wouldn't necessarily see a phenotypic difference. If you were at a location that did matter, you would see a difference. This forms the basis of a statistical test, and it's a statistical test that you can apply going right across the genome. So in this is the genome scan. Along the bottom we have location, and up the side we're measuring the strength of the association, the strength of that relationship. And we can see in this genome scan, we find that there's a location around the end of chromosome 15 that's highly significantly associated, and we call this a quantitative trait locus. So that's a useful tool. Inbred's a useful tool. We've seen that QTL mapping needs a genetically varying population. But then you might have noticed that, well, so if all individuals are distinct, then you lose the advantages of replicability. I was talking earlier about how great it is to have replicable populations, but we just created a population that wasn't replicable. So how to combine the advantages of inbred strains with QTL mapping population. So in the early 2000s, a group of researchers that include some of the people in the audience, Rob Williams, and maybe others, I can't see, proposed a type of population that would combine these advantages. So it would be a replicable population that you could do QTL mapping with. And what they proposed and what ended up getting developed is, so you start with 8 genetically diverse inbred strains. So these are inbred strains plucked from the sort of corners of the mouse phylogenetic tree, and accounting for a lot of diversity. That's the last line down there, more diversity than the human population. And then you breed them with each other. So through a series of kind of intercrosses, such that you get to a point where you have individuals that are carrying haplotypes from each of the 8 strains, so bits of the genome from each of the 8 strains. And then you set about inbreeding, and so you create the set of inbred lines where the chromosomes are these mosaics of the 8 inbred founders, and so each inbred strain is inbred, so you can get replicates from it. If you look across the inbred strains, you have genetic diversity and the basis for QTL mapping. So that's the collaborative cross, and there are a bunch of sort of derived populations you can make from that population. Oh, here are some papers that I've been heavily involved with regarding the collaborative cross. But there are other crosses that are related to the collaborative cross or derived from it that can be used complementarily with it. So one is the diallel. This is where you take the founders of the collaborative cross and you make them with each other, and you look at all of those combinations. Another thing you can do is you can mate collaborative cross strains to each other, basically make a diallel of the collaborative cross. We call that a CC recombinant inbred cross, or a CC RIX. Don't worry about the terminology. And then also a spin-off these three populations, I should say, these are all replicable populations in that if you do an experiment using them, you can repeat the experiment using genetically the exact same mice, or indeed you can do replicates within the experiment. A spin-off population was created from the collaborative cross called the diversity outbred cross. This is an outbred population, so mice are just mating with each other randomly, and the advantage of that is you get a lot of different combinations, but the disadvantage is that each mouse is distinct. So this is not replicable, and it's quite similar to an earlier population called the heterogeneous stocks, although I would argue, and so most people, that it's a bit better than the heterogeneous stocks. That's a very useful population, but I would say more as a complement to what I'm going to be talking about in this talk. So the collaborative cross is a type of multi-parent population. What happened, this is my interpretation of events, that a lot of people studying other model organisms looked at what we were trying to do with a collaborative cross and thought, yeah, I can see that's going to take a long time with mice, but we could make one really quickly in plants or flies or yeast. And that's what they did. And so there's a whole set of different populations in different model organisms, and they come under the heading of multi-parent populations. That's the generic name, and it's quite an exciting time, really, for model organism genetics and complex traits, because there are all these sort of new types of crosses, new types of experiments that you can do, and new types of derived crosses that you can make. Okay, so that's a collaborative cross and multi-parent populations. I'm now going to go through some G by T concepts. And as an example for G by T, because I think it's really useful to use concrete examples rather than talking about things in abstract. Let's think about drug treatment. So drug treatment, it's great when it works. You get symptom relief, disease reversal, but drugs affect different people differently. The formal name for this is heterogeneity of the treatment effect, or HTE. Some of the people call it treatment effect heterogeneity or treatment effect variability. It's actually a whole bunch of different names, but I'm just going to refer to it as HTE here. And what that means is that for some individuals, this variability of the treatment effect results in a lack of response, or even in some cases an adverse drug reaction, in other words a side effect, and that can obviously be a problem. And it creates a quandary really for regulatory bodies and for pharma companies and for doctors. If you have this drug that's got these known side effects for some individuals, what do you do? Because you know that you could be prescribing something that will harm vulnerable patients, but at the same time, if you hold it back, you could be holding back a very effective drug. So some kind of clue into what causes this heterogeneity is very useful. HT is partly heritable, which basically means HT is partly gene by treatment interaction. And there's lots of evidence for this. It's been well known for a long time. It is, however, really quite hard to study rigorously in humans. And I would argue that it's hard to study even in model organisms that are outbred, and people can argue with me about that. But this is, so G by T discovery is hard in humans for the following reasons. If we think about the standard genetic analysis, what does that do? Well, here we're trying to look at the relationship or look for an association between genetic state, which could be a SNP or haplotype or a strain, versus a phenotype that we measure. But in a G by T study, we're trying to associate the genetic state with a treatment response. So how is a treatment response different? Well, when we're talking about a treatment response, what we're really doing is we're talking about the causal effect of a treatment. And I'm going to dig into this just a little bit because I find it a really useful framework for thinking about these things. So let's imagine that we've got these two individuals, individual 1 and individual 2, and we give individual 1 the drug and we give individual 2 the placebo. And we measure the outcome for individual 1, and let's just call that Y1, and the outcome for individual 2, Y2. What I'm going to do now is I'm just going to label those with drug and placebo, and it'll be clear why I do that. So what we can then do is we can imagine a kind of parallel universe where at the point of the treatment assignment, individual 1 instead got the placebo. You can think of this as an unobserved outcome that would be observed if we were in that parallel universe. And likewise for individual 2, at the point of treatment assignment, we have this counterfactual where individual 2 received the drug. And so if those were the case, then we would be measuring Y1 placebo and Y2 drug. So the causal effect of the drug on individual 1 is the difference between Y1 placebo and Y1 drug. And the causal effect of the drug on individual 2 is the difference between Y2 placebo and Y2 drug. One thing that you'll notice with this is that something called the fundamental problem of causal inference, that the causal effect of treatment can never be observed. That's an important thing to realize for those of you who haven't thought about it that much. We can estimate an average treatment effect, estimate it, but we can only do so when 1 is very much like 2, when those two individuals are basically acting as counterfactuals for each other. So even though we can never quite get there when we're designing studies, that's what we're aiming for. G by T in humans is hard because it's going to be hard to match treatment regimes, disease severity, environment, genetics. There's obviously going to be ethical constraints in trying to do that. You can use twin studies and GWAS, but a lot of the time you're limited to sort of post hoc matching, and as a result you get quite low power. Low power is a feature of these studies that I'll talk about a bit later. So how can we try to embody this idea using a model organism, using mice? Well, what we can do is we can look at paired mice from a single strain, so same-sex litter mates, and we can give one. We can randomly assign one the treatment and one the placebo. And let's just say, okay, you've got individual I and you've got their pair, individual I prime. And we can come up with an estimate of the treatment response. Let's call it delta I, which is just YI drug minus YI prime placebo. And of course, if you're using inbred strains, you can gain extra precision by using more than one pair, lots of pairs perhaps. Moreover, if you have more than one strain, then you can start estimating the delta for that's inherent to strain 1 and the delta that's inherent to strain 2, and then you can ask, well, are those deltas different from each other? And on the basis of that, you can ask whether you have a gene by treatment effect, which is in this case a strain by treatment effect. And you can do that with more and more strains. And there, what that forms the basis of is a more general kind of linear model or regression model, where you can model the deltas of these different strains, and you can have an intercept, just like an overall difference, plus a genetically plus a genetically induced difference, and genetics here might represent a SNP or a haplotype or even strain itself, and that would be the gene by treatment effect. So by using deltas, you get this fairly simple model. In order to do this, of course, you need biological replicates and you need genetic diversity. And so how are you going to get those two things? Well, you can certainly get them from the collaborative cross. There are other populations you can get them from too, but this is what I'm talking about. So I'm going to talk about some studies that I've done following this path in the CC and the diallel. And these are all really focusing on this idea of modeling delta. So you design the experiment so that you can get delta, and then you fit a model for it. And so these are they're more coming down the pipe, but these are the studies I've been mainly involved in. I started getting involved in this in around sort of leading up to 2014 with a study on haloperidol. That was a diallel study. Another diallel study is on flu. There's quite a complicated pseudo diallel CC-RIX study that I'm not going to talk about on parent of origin. And for this talk, though, I'm actually going to focus on this series of studies, which are all quite similar to each other. And these are studies on therapeutic drugs that cause drug-induced liver injury, DILI. These studies published, 2 of them were published in 2017 and 2019. That's the tolvaptan and the idelalisib. The drug-X study should be submitted, I think, in the next few weeks. I'm not allowed to say what drug-X is, though, apparently. So until it's published. So these were all therapeutic drugs that got some way in the process, maybe even all the way through clinical trials before they started causing problems, and then the drug manufacturers came back and they wanted to see, what could we have done to try and catch these problems earlier. The general approach for these studies, so the objectives has been to identify risk factors associated with toxicity susceptibility, and also ideally to provide a mechanistic understanding of drug toxicity. And their general design has been to use 45 to 50 cc strains. That's how many strains we could get hold of at that time. And use 4 pairs from each strain. And these have all been done in one sex, but there's no reason why you couldn't expand it other than money to more sexes. And so we have replicates of the pairs. And
William, just to clarify, by pairs, you mean litter mates of the same sex?
Same sex litter mates. So each pair is the same sex litter mates, and they're randomly assigned the treatment or the placebo. Yeah. And then we're really careful about randomizing where in the course of time, because a real study can't all happen in one day. It's sort of spread over different seasons.
That's important to point out, because despite being isogenic, there could be maternal effects. So by doing it the way William just described, that's controlled also.
Yeah, and really, I mean, you don't. I guess I used to think of these things as like rules that you had to abide by, but I think if you understand the nature of the causal question, you don't need rules. You can go by intuition. And so we measured various biomarkers of liver injury and used those to do QTL mapping. We also did the same for gene expression in 2 of the studies. I'm going to talk a little bit about the results, but I'm not going to really do a deep dive into them, because there's something else about this that I want to talk about. So tolvaptan, it's a drug that was used to treat polycystic liver injury. If we look at ALT, which is a biomarker of liver injury, if we look at that across the strains, we found that there were 3 strains where there was a really big difference between the tolvaptan and the control. It's not a lot of strains for that to be different, but it was a really big difference. We did our QTL mapping model by using delta as a response, and we didn't get something super significant, but in terms of the other evidence we got, it was really, really very clear. And certainly looking at the strain effects, it was very, very clear. And I'm going to talk about power in a minute. And the other study was idelalisib, which was used to treat leukemia. That we had repeated measures outcomes for that, so we had a bit more power, and we found multiple QTLs across a range of biomarkers. Those are the things we talked about in the paper. Something that we didn't really say very much about, because it wasn't of immediate interest to that audience, was the thing that I found really interesting. And that's what happened with the eQTL mapping. So what I'm going to show you is, I'm going to show you the eQTL mapping that we did for drug X, but the same thing happened with the tolvaptan study. So this is an eQTL mapping plot, and probably some people are familiar with this kind of plot, but others, it just looks like a complicated picture. So what's going on here is that on the vertical axis, we've got the location of the transcript that we're measuring the expression of. And on the horizontal axis, we've got the location that we find a QTL for that transcript. And the red line along the diagonal, those are all the cis eQTLs, places where we find a QTL in the same location as the transcript, which you would expect there to be lots and lots of cis eQTLs, and then places off the diagonal are trans eQTLs. Okay, so this is, anybody who's done eQTL mapping, this is sort of a common looking plot. We've got this big diagonal and we've got things off the diagonal. The colors represent the significance of the hit. This is just for the placebo treated mice. Okay, and let's look at the drug treated mice now. So we see a similar picture for the drug treated mice. So this is just QTL mapping on the drug treated mice. So what happens when we map on the delta? When we map on the delta, we saw this, and at first I was a bit sort of shocked. I wondered whether we'd done something wrong because it doesn't look like the usual kind of QTL mapping plot. It doesn't have a diagonal, right? And the more I thought about it, I realized, no, actually, this is probably what you want. This is probably something that's telling you you did the experiment and the analysis right, because all of those cis QTLs, they weren't really affected by the drug, and so the picture is going to be sparse. The number of hits. Somebody's asking a question: what's going on at distal chromosome 12? I mean, not quite sure. Well, yeah, ask me later about that. So yeah, so we see a very sparse picture, and we don't see the hallmarks of a QTL plot in terms of the diagonal. And this really leads us to some observations about delta. So if we were to have studied treated mice only, we wouldn't have realized that the treatment response that we thought we were measuring is actually completely confounded with the baseline measurement, and that the results largely would have reflected those baseline effects. So we wouldn't really be learning anything about G by T, we'd just be learning about G. The delta values tell us about the treatment response, the actual response to the treatment. The other thing we notice is that the genetic effects on treatment response are few. And not unrelatedly, they're detected with relatively low power, and they're estimated with less precision. So let's try to understand why this is. And if we think about modeling a placebo phenotype, the way we might model that would be as, okay, there's some overall mean or intercept, there's the effect of genetics, and there's some noise. If we think about modeling a drug phenotype, then there's some different mean, genetic effects, and there's different noise. When we look at delta, what we're looking at is the difference in those means, which is what we would call the overall treatment effect. We're looking at the genetic effects on delta, and we're looking at noise minus the noise. So the drug noise minus the placebo noise. Now, if you know about random variables and variance, then you know that noise minus different noise is not less noise, it's more noise. So the noise of the delta is the noise of the placebo plus the noise of the drug. So what that means is that the delta is much noisier than either the placebo or the drug. And so more noise means you have lower power, and this obviously has implications for experimental design. The good thing about it is that using the delta avoids having to separately model drug noise and placebo noise. It it also does not assume that those two noises are the same, as would be the case in a traditional interaction type G by T model, which I'll talk about briefly on the next slide. It's simpler to fit, to diagnose and test significance. So I mentioned that thing about a traditional interaction model, and I'm just going to go through just a little bit of math. So a more traditional way to model gene by treatment effects is what I call the interaction model. So modeling G by T as a statistical interaction. So here we would have a regression where you've got the intercept and then a term for the treatment effect, a term for the genetic effect on the outcome, and then a gene by treatment interaction. And that's the kind of model that people usually use. And it sounds good because it's an interaction model for looking at gene by treatment interaction. So they both got the words interaction. But one of the assumptions about it is that you are assuming that both types of individuals have the same type of noise. And if you're used to looking at treatment data, I mean, what I've seen so far is that the treated individuals are often a lot noisier. It's sort of interesting to see how you can relate that G by T interaction model to the delta model. And you can do that simply by taking the formula up there and writing it out for a placebo-treated individual and a drug-treated individual, subtracting one from the other, and you get what looks like the delta model, except that the noise term is the drug noise minus the placebo noise. So, okay, yeah, that's all the math for now. So I'm just going to briefly talk about two of the other studies that we've looked at using this. So the first one, which is really where I got into thinking about gene by treatment effects, was using haloperidol, which is an antipsychotic, which is extremely effective antipsychotic, but it has this horrible side effect that afflicts some people called tardive dyskinesia. What we did here was we used a diallel. This is where we crossed the founders, in this case of the collaborative cross, with each other. So all of these different combinations use the same kind of like same-sex littermates, and this is a picture where we've colored the intensity of the phenotype, that so how high the phenotype is by shading it per cross. And this is actually plasma haloperidol, which isn't really a delta. It's only defined on the treated. But what we can see is that if your mother is an NZO, you're going to have a much higher plasma haloperidol level, and so we can see a strong parent of origin effect there. If we look at something where we do have a placebo and treated phenotype, and therefore we can get a delta, this is activity in the open field, which is sort of a measure of anxiety. We can see that there's a different kind of pattern here, some interesting maybe parent of origin interactions. We worked out a fairly complicated model for modeling different types of effects on this, which you can read about, but that was the paper where we'd previously looked at sort of ordinary phenotype diallels and thought thought that moving to a treatment effect would be straightforward, and actually it opened up a whole new series of questions and a new thought process. Another study was by my student Paul Maurizio. This was on influenza A, and here the design of the study explicitly took advantage of knowledge about the fact that the treated individuals were going to be more variable than the mock individuals. What this was looking at, we were giving the mice influenza and tracking over a period of 4 days how much weight they lost. But we're also doing this for mock animals who were going through the same kind of experimental process, except for the flu. And so as an experimental design component, what we did was we used 3 treated mice to 1 mock mice. And you can see in this plot, as you get to day 3 and 4, you actually have a lot more variability than in the corresponding mocks. So I'm probably not going to explain these pictures, but if you know about diallels, then they might mean something to you. So design is obviously important, but power is obviously important, particularly when you know that using these kind of this delta idea, you're going to be in a more low power situation to start with. So that means you need to power your experiment carefully, and something that we did fairly recently, well, last year, was really do a deep dive on, okay, if you're going to use the collaborative cross for these kinds of experiments, really for any experiment, and especially if you're going to use it for QTL mapping, you really need some numbers on, like, how many replicates do I need in order to get good power. And the outcome of this really is that that compared with an outbred population, the collaborative cross is extremely efficient. You know, once you've got sort of 5 replicates for 50 strains, you're not talking about a lot of mice, and you've got pretty good power to find QTLs. One of the other interesting things to come out of the drug liver injury studies is the ability to use the shared controls for a sort of a rigor and reproducibility study. So in each of these different drug studies, we collect control mice, and you know what they go through is not exactly the same between the studies, but it's still fairly similar. And what we've been doing is seeing, you know, how much drift, how different are the baseline sort of QTLs and so on that we get. So that's another real advantage of replicable populations, that you're continuously adding to this bank of information.
Will, on your power analysis in the upper right corner there,
yes,
that is a % genetic variance, % total variance?
So I've got some extra slides on this, because I thought you probably would want to ask about it, and ask me at the end.
Okay.
Which is very soon. Oh, the end. Not quite yet though.
Should I ask you now?
No, because ask me at the end of the end.
Okay.
So gene by treatment interactions, what I described was a systematic model organism-based approach to G by T discovery that uses genetic diversity, biological replicates, analytic strategies, and it doesn't have to be using the collaborative cross. I think the ideas that I've described would apply to other populations as well. the resources I talked about were the collaborative cross, the diallel, and these other kind of derived populations from the collaborative cross. I think the take-home message that I get from this, which I want to pass on to you, is that when it comes to gene by treatment interactions, good statistical modeling is important, but more important is good design, and that replicable multi-parent populations are a good place to start. And I'd sort of played around a bit with different sorts of causal modeling on outbred populations before I'd started thinking about this, and the design piece was just more and more kind of seductive to me, and it reminded me of this paper from 2008 for objective causal inference, design trumps analysis. And so this is really arguing that this is in the Annals of Applied Statistics, which is a very well known stats journal. And what it's really arguing is you can come up with fancy statistical modeling, but you know, the real bottleneck is the design of the experiment. This, by the way, was a time when this is Donald Rubin, and this was a time when the word trumps and Donald could be used without thinking of a particular person. So the other person who I think has been a real thought leader in this is Alan Festing, who is in the field of toxicology, and he for a long time has been advocating for the use of using panels of multiple different inbred strains in toxicology studies. The importance of both of those things, that there should be inbreds, but also multiple inbreds. So a shout out to him. These are my acknowledgements. I want to thank these people, in particular from my lab. Yanwei Kai has been involved in all of the drug liver injury studies, and Mary Mosdale is my prime collaborator, who's really been a pioneer in that work. You can learn more about my work at the website down there. And yes, I'm happy to take questions. Thank you for your attention.
Clap, 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 clap.
Yes, I guess should we just start rolling through the chat questions first, and then at some point after that, we can just transition to the more informal Q&A. I think we don't have very many questions, so maybe we can just start and people can ask the questions themselves in the chat, starting with David.
Yeah, so when you had the two transcriptome experiments, the eQTL experiments, they looked like there was a really strong trans band on the end of chromosome 12. So basically lots of eQTL mapping there. And I wondered if you know if there's any reason for that, like this one. So if you look at sort of distal chromosome 12, there's a lot of eQTLs that map back to there. And I just wonder if there's any reason.
We didn't really find, we did look at that. We didn't have like a great story for it, but we did spend some time looking at that, and also kind of quantifying how significant these trans bands are. I think trans bands are just really interesting, actually, and yeah. I don't think they're talked about enough in human studies. I know that it's quite common to use these sort of PCA like peer factors and correct gene expression data for them, and that unfortunately has the it seems to have, you know, I might be misunderstanding something, but it seems to have the effect of removing the trans bands. In these studies where I trust the design a lot more regarding the possibility of experimental confounders, yeah, I find the trans bands interesting and understudied, but I don't have a specific kind of clincher
David, it doesn't change anyway. Why? So the trans band that appears on chromosome 4 in the drug treated is the obvious interesting one. There's also one on distal 1 that is only seen in the treated side. So those seem much more interesting. But William, I guess what I would say is what's doubly or triply interesting is the cis eQTLs, the location of the trans band on 4. So is that how you were led to your candidate gene on for that trans band?
On 4.
I think
on 4 for the drug treat.
It's even more obvious.
So oh yeah, this one. Oh yeah, this trans band. Yeah. So this is a very clear trans band, and it's very clear what marker it's on, but we did not kind of figure out any particular reason that might be driving it, but it was a very clear trans band. Yeah.
But on a plot like that, it would be cool to put an outer circle around those spots that are on the cis diagonal, because those are doubly important.
Oh, I see. Yeah. Mm-hmm. Mm-hmm.
Yep.
Yeah.
Okay. I'm done.
Okay, so I think then the next set of questions are going into the math. So I guess, Sonic, do you want to start or comment?
Yeah, hey, Will, great talk. Enjoyed talking to you. So you know, you mentioned that the delta should have bigger variance. I'm not sure I would necessarily agree with you because there is a shared genetic variance that will cancel out when you take the delta. So, you know, it's like a paired t-test, right? In a paired t-test, you know, your delta is essentially the equivalent of a paired t-test. And in your there, what you...
So the delta, it's not that the delta has, I'm not saying that the delta has bigger variance. I'm saying that, I mean, I might have literally said that, but what I was meaning was that the noise of the delta was greater.
Yeah, of course, it depends what you mean by noise. In the third equation that you have, the genetics, which is labeled as genetics, is really delta genetics too. And that, it depends what you mean. Of course, as you say, the pure noise is certainly greater, but the genetics noise is smaller. And depending on how you are analyzing it, it depends like what your noise term is. You know, when you're doing QTL mapping, you're looking at a particular locus. The genetic noise is part of your noise in the error model. So in that sense, anyway, so that's one thing I want to point out, or maybe it's a matter of semantics.
It might be a matter of semantics. I mean, in this, so I say genetics, but I don't. In this kind of simplified, I go into here here, maybe it's easier. The genetics is not the same. So really it's more like this equation here, the interaction model, ending up with
so, but your model does not have the background genetics in it. Like, you know, the all, you know, you're looking at a particular locus, but there are all these other loci on other chromosomes that are contributing variants. And when you take the delta, that will cancel out.
I want it to cancel out because I don't want, I want to focus just
but
on...
but the G by E will not cancel out. Or G by T will not cancel out.
Yeah, I want to get rid of I want to get rid of the shared genetic, well, what I think you're calling the shared genetic component, because I don't want to do a study that is focused on treatment response that ends up just telling me about the baseline phenotype. So I want to use the design to mean that I don't have to model as much. And
so I totally endorse the delta. I'm for it. It's very good. What I'm saying is that I don't necessarily agree that the delta has low power. I would argue that the delta might actually perform quite well if there's a lot of you know, if the genetic control or the background genetics.
Oh, I think I see. I see. So do you mean you're saying that the delta would be powerful compared with testing for G by T in an interaction model?
Using the model that you have there, yes, I would agree. So
using the top model.
Yes,
yeah.
Because, you know, what it does not have, it does not explicitly have a term for the shared genetic variants,
which you're saying is kind of burdensome from a statistical perspective, and that
right.
And having that term would lower power because you're having to model
you're making fewer assumptions by taking the delta. I'm all in favor, you know. Okay, the other point is not really point, really. It's a comment. Is in your you showed us that the eQTLs for your delta had far few or very little, if any, you know, cis effects, and you know, I just wanted to comment that at least in one study that I've been involved with, that it was not the case. Obviously, it was a very different kind of study. It was in Arabidopsis, and the drug was water. You know, so it's
okay. That's
but you know, whether you give wet or dry, so it might be that the biology is quite different. But in that one, we did not see the. I'll put it in the chat.
Okay, thank you. Yeah,
and we use the delta that you find, you know, the same kind of.
That makes sense, Shonak, because in Arabidopsis, that's going to be a highly polygenic environmental interaction with the genome, whereas in this case, it's one specific manipulation that's relative, that you might think would be multigenic or even oligogenic?
Right. I mean, yeah, water has much more, has global effects, whereas
Yeah, right. Yeah. Yeah. No, that's a really good point. Something else that I don't talk about here is the transformation, and so I talk about what is that, modeling. Let me find the best slide to talk about it. Yeah. So if we think about the way we're describing the delta, actually, delta can be correlated. Delta can be correlated with baseline and drug, or you can induce a correlation by picking the wrong transformation of Y. So in the gene expression, for example, Y, what I'm calling Y, it was the log of the gene expression, right? So YI was the log of the gene expression for the treated mice, and YI prime would be the log of the gene expression for the placebo mice. And so the delta is the log of the fold change. If, however, I had not logged, if I had just done the gene expression, YI is the gene expression unlogged, very, very non-normal distribution in most of those cases, then we would see that the delta and baseline, they're correlated with each other. So it's kind of a subtle point, which if I had a whiteboard, it would be easier to explain. But you can. Is the annotation tool enabled? Okay. Am I allowed to annotate the screen?
Can you? I mean, you can try. I'm not sure. I'm looking to see. I don't see any option. Is there an option for that? I don't see an option for.
Yeah. Sometimes on Zoom, something I find quite useful on Zoom is I can kind of scribble over people's slides. Anyway, if I was able to, I would draw a situation where you have the drug and the placebo, that they're increasing in kind of a non-parallel fashion, but the increasing distance between them is an artifact of a mean variance relationship.
Okay, so Carolina and Byron, are your questions answered by that conversation?
Oh,
not quite.
Yeah. So
go ahead.
Is that Byron?
That's me.
Yeah.
I'm not Carolina.
Yeah. not quite the lightness of tone of voice I was expecting. So yeah, we've looked at that, and yeah, I mean, I think there is value in adjusting using ANCOVA, basically incorporating, so not only doing the delta, but then also including, what would it be, YI placebo into the model that you're fitting as a covariate. So basically correcting. So that's in particular something that we've we've been using in other projects when it's not easy to find a transform of y whereby delta and baseline are correlated. If we can't decorrelate them using a transformation, we'll use a covariate to just kind of ensure they're not correlated. Yeah.
Yeah. Thank you.
Okay, great. So I see we have a grad student question. So I think this would be a good time for me to stop recording and move more into the informal Q&A session, so we can maybe expand on like some more like fundamental details if students and other people are interested.